Welcome to the tutorial for Pymol. Pymol is an abbreviated term for Python-based molecular visualization tool. Pymol is a multi-platform software. It is used for graphical representation of molecules such as protein, enzymes, DNA, chemical compounds, etc. Pymol is a graphical user-based, that is, a GUI-based software, and it supports command line options also. So once we get familiar with Pymol, you may write your own Python scripts to interact with Pymol. Hence, it is an easy to use open source software. It is feature loaded. With the help of Pymol, we could predict and study the active site of our molecule, or we could also measure the hydrogen bond lengths, the torsional or the dihedral angles of a protein molecule. This is a very important aspect which we are going to see in this Pymol tutorial. The other thing that we could do with Pymol is measuring the hydrophobicity or hydrophilicity of the molecule. It could be done by generating the electrostatic potentials. Hence, Pymol is not merely a visualization tool, but one could also easily use the features which are there in Pymol. To download Pymol, you may, in your web browser, go to this particular website, pymol.org. It is maintained by the group Schrödinger. Since it is downloaded for Windows, Mac OS, and Linux, we could install it in any operating system. Once we download the exe installer in Windows, we could successfully use Pymol for visualization and analysis purpose. The input format for Pymol is actually a Pdb data file. Pdb stands for Protein Data Bank. Let us see what Pdb actually is. The Protein Data Bank file format is actually consisting of the sequential, the structural, and the annotation information for a particular protein molecule. Each PDB is identified by a particular PDB ID, that is, a PDB code. This PDB ID is a combination of 4 words or 4 letters, one of which must compulsorily be a number. For example, we have several PDB IDs. One of the most commonly used PDB ID could be that of protein enzymes. Such enzymes, one which we are going to work with is 3-PTB. 3-PTB stands for trypsin enzyme. As you can see, there's a number there, and we have other 3 letters. Another code which could possibly be 1U19. That is an inactive conformation of the rhodopsin molecule, which is used for photoreception. So in this tutorial, we will use 3 PDB codes, 3 PDB entries, and those are 1U19, 3-PTB, and 3-CAP. So these are the 3 PDB entries which we are going to use for this particular tutorial, and I suggest you to download these PDB files in the Pdb file format. Although we could use any other file format because there are varied amount of type of file format which are supported by Pymol software, but we will preferably use the .pdb extension, right? Okay, so let us now first download one of the PDB files, that is the trypsin molecule. 3-PTB is actually the trypsin molecule. It is highlighted here. So the trypsin molecule is actually the active form of trypsinogen. Trypsinogen is a digestive enzyme. It is found in digestive system and catalyzes some of the chemical reactions, enzymatic reactions. So in the PDB itself, we could just analyze what this molecule or entry is about. So scrolling down, we could see that in the macromolecular section, the name of the molecule is beta trypsin. It has chain A. They could be chain A or chain B, but nevertheless, we have the chain A here. The amino acid length, the sequence length is 223, and Bos Taurus was the organism from which it was purified or crystallized. So this is the general introduction for this molecule which we are going to work with. If you want to see the ligands which are interacting with the molecule, we could see two ligands here. The first one is the benzimidine. There's a 2D diagram drawn in the PDB site if you go to the 3 PDB section. And another ligand we have here is the calcium ion. The calcium ion is also present in chain A. Now, what I suggest you to do is you could download this PDB file in the PDB file format, right? You could click on the download files option, and from there we have a varied amount of sequence format. As you can see, there's a PDB format which we would need, the FASTA sequence, and also we have the MMCIF format, or below we have the structure format, also the CIF format. The structure factors are represented, but we will download the PDB file format. So our file is downloaded here, and the next thing we could do is we would in the search bar enter the term 1U19, that is the code for the rhodopsin molecule, the inactive state. And similarly for the rhodopsin molecule, what we would do is we would click on download files option after reaching there. So we have reached to 1U19, which is actually the crystal structure of bovine rhodopsin. It is being the resolution is 2.2 angstroms. And for this bovine rhodopsin, we are going to download the PDB file format, right? So we would perform the similar options. Rhodopsin is actually like giving you just a brief description. So rhodopsin is a molecule which is related to the opsin protein. So the opsin protein is not a complete protein, not a holoenzyme, right? So what happens, like opsin binds with 11 cis retinal, and therefore after binding with it, it becomes an inactive form. So this 1U19 is an inactive form. The active form is 3CAP. So 3CAP is not bound to its ligand. As we can see, it is the crystal structure of the native opsin. That means the rhodopsin is in its ligand-free state. The native opsin we have, so it is in ligand-free state, right? As it is mentioned here, it does not have any ligand. And what we could do is possibly we downloaded this file, and similarly we would do it for the earlier one also. So we have both of them, right? The active is already downloaded, and now we are downloading the inactive form also. So below we can see we have downloaded all the 3 files which we would further need in our analysis purpose. We have the 1U19, which is actually the inactive conformation. 3-cap is the active conformation, right? So the 3-cap is the active one, and with both of them what we are going to do is we will load both of them at one go in Pymol software, and after that we would superimpose them upon each other and then see what are the conformational changes which are occurring by the purpose of ligand binding. So what happens upon ligand binding? How is the conformation changing from 1U19 to 3-cap? How is it happening? What is the actual measurement with the help of which like the conformation is changing? We would analyze that, right? And with the help of 3-PTB, we would get to work with some active sites, etc. So first of all, I have created a folder in the desktop where all the information or the file formats is stored. We do not need this active and inactive. So what we would do is we would click on 3-PTB and open it with the Pymol. So click on open with option and open with, choose another app and then Pymol. So this is easily available. I think everyone could do this. So this is the first option, and it could be loaded as seen on the screen here. So this is the default screen, which is actually there when we load the molecule, right? So this is our molecule. It may be quite messy at first, but as we proceed with this study, it would become quite familiar to you. So this is the first option, how we open the file. Otherwise, you could also use the file open option, which is there in the command line window, the upper window we have here, right? So this is the file open option, and you could then direct yourself to your current working directory, the folder Pymol, and then open it. It would open it as usual. Okay, so these were the two things, like two ways how to open a PDB file. And as you can see here, there is a command input area also. So we have two windows. The below one is the display window or the work area, and the upper one is the command line window, right? It has certain menu bars and the command input area with the help of which you could interact with. So there's a third option also. You could place a command. You could type a command in the command input area, which is called load and then the file name, right? So before doing this, you have to ensure that you are in your current working directory. You have to direct yourself to the directory, right? And then after that, here it is showing that it is C users HP desktop Pymol. So it is the path of the current working directory, and the molecule is being loaded as 3-PTB. Okay, so first of all, let me tell you about the interface of Pymol, since we are going to work with Pymol. So the interface of Pymol is quite a simple one. This all text which is written here is actually the default text. Whenever any file is being opened in the Pymol, it is being written as all. And when we click on it, it could hide and unhide. And similar things could be done for the 3-PTB tab. So clicking on the tab will hide or unhide the display of the protein molecule, right? So we have this display area where the protein is being displayed, the black surface, right? On the right side, we have this play area where we could play with our protein, perform some actions on the protein, right? And we have certain buttons to interact with. These are 5 buttons here. Like on the right side of the 3-PTB tab, we could see certain buttons, right? So the first button is the action, and then show, hide, label, and color. So these are the 5 options we have here, right? The action button has many sub options. For example, zoom, orient, center, origin, right? So these are the many of the options which we would be using. So these are available, and what happens if I drag it to a certain position and then I click on action, zoom, it would revert back to the original position, right? So we have certain actions we could perform on the protein molecule, right? We have another actions also. Now we would use them further. The second button is the show button, right? What happens here is the line structure currently, and if we want to change the display, we could display it as lines, sticks, ribbon, cartoon, etc. So we could easily change our option or display, and therefore the current display is actually a line model, and it is quite messy at first. So we could change the display to maybe say a sphere. So when we change it to a sphere, so we could see certain balls here, and each of the color represents an atom. For example, red color represents, the red ball represents oxygen. Now we would click on hide everything, and after everything is hidden, we would want to show it as a cartoon model, right? So once we show it as cartoon model, I think now it is better than before. So but it is green in color, and we could make it visually more interesting by clicking on the colors tab. Now we have the colors tab here, right? The protein is now being colored with a particular color scheme. So in the colors tab, what we could do is color it by element, right? So the default is the CHNOS, We could color it by chain, but because it has only one chain, so the color will not be changed. Only chain A is there. Now I would like to color it by secondary structures. So the secondary structure what we have here is helix sheet loop. We could also color it by a spectrum. It would represent it as rainbow. So once we color it by spectrum, it would be represented as rainbow, but I think this is also okay. Let me show you how to represent it as a rainbow colored. Click on the colors tab, and afterwards you could color it by the rainbow scheme. So here we have the colors tab right here after the label tab, and once you click it, we could see it as rainbow. The current visual color scheme is the alpha helix beta and the sheet. So the green color represents the loop, the red is the alpha helix, and now the color is being changed. So we could direct it by the secondary structures. So that is more relevant, right? Okay, let me rotate. So yeah, how is this rotation actually happening? How am I operating this molecule? So let us talk about the third panel which we have here, right? The molecule is being rotated. The third panel which we have is the mouse panel, right? How do we operate that protein? So I suggest all of you to pick up your mouse, and then when you click the left button, you could rotate the mouse. Like you have to long click, press slightly on the left button, and then you could rotate your molecule, right? It is written right there in the bottommost, the right bottom portion. Okay, so I suggest you to click left click on the mouse and then try to rotate your molecule like this. So it is being rotated. We have other options also. You could zoom in or zoom out to your particular molecule. For example, this could be done by the right click. Similarly, you need to right click on that particular molecule. Okay. And after right clicking, what you need to do is you need to actually move your cursor towards you. Okay. So this is the rotation thing, and when you right click, and you have to move your cursor towards you, and when you move your cursor towards you, it would be zoomed in. Okay, so let's try this once again. You have to right click on it and then zoom in. So when you move your cursor towards you, it would zoom in. Otherwise, inside, if you place the cursor inside, it would zoom out. So this is zoom in, and right now we'll place it inside. So this is zoom out. So likewise, you could play with your molecule, and this molecule could be played with. This could be rotated or zoomed in or zoomed out, right? So if I want to move my molecule a bit to the left or to the right, what I would do is, let's see. We have another option. I would drag it, and this could be done by clicking on the wheel, press the wheel, and then drag it to the position. I could drag it to the left, or then I could drag it to the right also, and I would probably like to place it in the center. So after placing in the center, so this is how we play with our molecule with the help of our mouse. The last option which we have here is the wheel, and we could slab our molecule. This is used for hiding and for unhiding purposes. So hiding and for unhiding. This could be useful for further studies. Okay, so this was all about the rotation, the movement, the dragging, and the slabbing of a particular molecule. Okay, so now let me make you a little familiar with the principle of Pymol, how it is actually working. Right? So for this, what you need to do is, you need to direct yourself to your current working directory. In the current working directory, what you need to do is, just minimize this. Yes, you need to right click on 3pdb.pdb, and then you need a decent Notepad++ software. You could use any notepad, but this is preferable. So you need to edit it with Notepad++, open it, and then let's see what is the Pdb file format. So after scrolling down, you could see this is the Pdb file format. This is a serine protease enzyme, and It's a serine protease, and the molecule name is beta trypsin. The chain A is there, and we have the enzyme commission number. Similarly, when you scroll down a bit, you could see atoms and the molecules. So what we have the first amino acid as? So we have this amino acid as isoleucine, right? And for each atom of isoleucine, we have certain numbers written, right? Let us see what are they. So isoleucine is the 16th amino acid which is there in the residue in our protein, and for that 16th amino acid we have certain numbers written here. For example, in the first line we have here is minus 8.096, 9.599 and 20.309. These numbers suggest the x, y and z coordinates of that particular protein molecule. So what happens when the protein is being crystallized in the X-ray diffraction study? What is the data that we actually get? So the data we get is the x, y and z coordinates, and Pymol actually uses this data, processes it, and then transforms it into a visual representation. So that is the basic principle of Pymol in brief. So I hope you get the principle of Pymol, how it is actually working. And this is done for each and every amino acid residue and each and every atom. Right, so this is the file format and the information that Pymol is actually using. Okay, so these atoms are not of very importance to us, but to software these coordinates are quite important, and that is what is helping the software to reproduce the protein structure from the coordinates. Right, so that is the overview of the principle of working of Pymol. Now what we could do is come back to our original molecule, and first of all let us just display the sequence, because it would be quite easier to work with if we have the sequence here. So you need to go to the display control and click on display. After clicking on display, select the sequence. So we have the sequence here, as you can see. So the sequence is actually, see, the first one is the Ben, that means the benzimidine. Remember earlier we just said that PDB entry had a small molecule benzimidine attached to it. And for our protein molecule, what do we have here is the first amino acid is isoleucine, and then we have valine, glycine, and then another glycine, tyrosine, tryptophan, etc. So these amino acids are represented by the one letter code and are colored according to the color scheme which we have defined, right? Okay, so let me just select isoleucine, and once we have selected, we could zoom it a little bit so that the isoleucine is visible, but it's not quite visible. So what we could preferably do is we would change the settings, and we would not want to show it as cartoon, but we would represent it in the form of a line, right? And this could be done with the help of the buttons which are provided at the right side of the select tab. Remember, you have to use the select tab and not the 3PTB or the all tab. So in the select tab, we need to hide the cartoon representation, and then we would show it as lines. The color is actually not nice, so we could color it by, say, blue. Okay, and we would also like to label the residue, what is the name and the amino acid number. So we could go to the label tab and label the residue. So as you can see, this is isoleucine and this is the 16th amino acid. We could also label the atoms and go to the label button again, and then we could select this and then go to label button and select the atom name. So the atoms will now be selected. Let me zoom a little bit. Okay, so the atoms are quite visible. We have the atoms such as carbon, oxygen, C alpha, nitrogen, C beta, C gamma 2, C delta 1 and C gamma 1. So this is the molecule, the isoleucine amino acid residue, right? Similar things could be done for valine. So what I wish to do here is for valine, I would just like select the valine with a similar procedure what we followed in the case of isoleucine. So valine is the second amino acid which is visible from the sequential order, and I would select valine and then color it by some coloring scheme, say pink, and then measure the distance between the atom, a certain atom of valine and another atom of the first amino acid. So likewise, we could actually measure the distance between the first and the second amino acid. So I have just used the action button and then zoomed out. So for valine, I'm doing the similar things. I have selected valine, and then I would hide the cartoon, okay, from the hide button. And then afterwards, what we could do is we would show it as line. So let's do that. And now it is being shown as lines. Let's color it by pink color. So I think hot pink will do. Okay, so now it is clearly visible, and we would label it as, we would label the residues, right? So this is valine, which is the 17th amino acid, and let's now label the atom names also. So the atom names are there. Let me zoom in a little bit. Okay, I would drag it, use the mouse cursor. So these are the atoms of valine, and I think the peptide bond is quite clearly visible because we have the linkage between the N and the C. So we have the nitrogen atom of the pink colored residue, the valine, and the bond between the carbon of the blue colored residue, that is isoleucine. So the bond is known as the peptide bond, right? So the peptide bond is quite visible. So what we could do is we would now go to our command line window, and we see a menu bar option here, which is the wizard option. We will now go to the wizard option and go to measurement, right? So what are we going to do? We would go to measurement, and then we are going to measure the distance between a certain atom of amino acid residue, valine, and another atom of isoleucine, right? So after clicking on measurement, what do we have here? We first need to select two atoms. So the first atom we need to select here, it is written here, please click on the first atom. So let me select the first atom as oxygen of valine, and the second would be probably C gamma 1 of isoleucine. So let's do that. So we would now first select the first atom. Okay, we could measure the distances here, and then we need to click on the first atom, which is the oxygen atom. After clicking on the first atom, it will show that click on the second atom. So now we need to click on the second atom, and the second atom will be C gamma 1. So the gamma carbon. As soon as I click, I see a number here, the numerical digit written here. It is 4.2 angstrom. I could color it by say cyan. So let's go to the color tab, and so it is now clearly visible. And okay, so the distance between the oxygen and the C gamma carbon of valine and isoleucine, it is written as 4.2 angstrom. So quite easy, isn't it? So we could measure the distances between these atoms. So let us now measure the angles between the measurement. And for the angle measurement, as we know, we need 3 atoms, right? So we have a variety of distances. We would now measure the angles. So the 3 atoms would be probably the nitrogen of valine, carbon of isoleucine, and oxygen of isoleucine. So let's now select the first, second, and the third atoms. So the first atom is, you just need to click on the first atom as nitrogen, the second atom as carbon, and now as soon as you click on the third atom, oxygen, you would find an angle, a value. See, the value is there. As soon as we click, I could zoom it a little bit. So this is 123.4 degrees. So now we know the angle which is being subtended by isoleucine and valine, the angle between the NCO group is actually 123.4 degree. So likewise, you could perform some useful analysis, and this could be easily done with the help of wizard and measurement tool, right? So this is as easy as that. We could measure dihedrals or distances and angles between those atoms. Okay, so this was all about the hydrogen bond measurement, and next what we would do is we would see what are the regions of hydrophobicity and hydrophilicity of a particular molecule. Okay, so first of all, we would delete all these measurements. Okay, we would delete the measurements. We do not want these measurements for now, and we would click on done. So the measurements will be deleted. And after we do this, we will center our molecule. So it is being centered here, and I suggest you to open the action button on 3pdb tab. So clicking on the action button of 3pdb tab, we have a variety of options. Find the option of generate, and when you find the option of generate, click on vacuum electrostatics. So after that, you see only one option, the protein contact potential. So just briefing you a little bit about protein contact potential. So the name itself suggests for each interaction which is being there in the protein, for example, the electrostatic interaction, what the software does is it calculates the energy, the potential energy of that particular contact. Hence the name protein contact potential. And as soon as you click on generate vacuum electrostatics protein contact potential, what the software will do is it will for only the electrostatic interactions, the polar interactions, it would calculate the potential energies. It could be in positive or negative, and we would have an index value, and the surface of the protein could be represented in the form of hydrophobic regions and hydrophilic regions. So you need to just click on protein contact potentials, go to action, and then you need to select the generate option. Okay, so we have this protein. Let's repeat that. So we need to go to action, click on generate vacuum electrostatics, and then protein contact potential. Wait for 1 or 2 seconds and the contact potential will be generated. So as we can see, the protein contact potential is being generated here. So okay, so you can see here 3 colors, red, white and blue. And what are these colors actually representing? Let's see here. If I rotate the molecule a bit, I could see the colors here. So I see the regions of hydrophobicity and hydrophilicity. And how do you identify the regions of hydrophobicity and hydrophilicity? Look at the index values, the numerical values which are written here. One is on the negative hand side and one is the positive values. So the negative values, the red colored regions are the regions which are hydrophobic in nature. So that's why they have a negative energy, the negative electrostatic potential. On the other hand, the blue colored regions signify the hydrophilic portions. Okay, so these are the hydrophilic and hydrophobic portions, and the middle portions, the neutral portions, are whitish in color. Let's rotate our molecule and see what are the regions of hydrophobicity and hydrophilicity. So as you can see, we have a lot of shallow and deep grooves inside our protein molecule, the surface representation. And you could zoom in and find certain cavities, binding pockets. So after we have our hydrophobic and hydrophilic sites or regions of the molecule, what we could do is apply the principle of like dissolves like. For example, if I tell you a hydrophobic molecule will always tend to interact with another hydrophobic molecule, right? So we have our benzamidine group, and it would probably like to have a hydrophobic binding pocket, right? So our aim is to find or detect a hydrophobic pocket from this electrostatic potential. So a hydrophobic pocket, if I zoom in a bit, we do not see any regions. We have hydrophilic regions, and these outer surfaces are actually what are interacting with the water molecules, because water molecules are hydrophilic in nature, right? So the outer surface are interacting with the water molecules, and the water molecules are making the protein stable in the cytoplasm of a particular cell. So that is how the whole environment is being done. If I rotate the molecule, I see certain patches of hydrophobicity. In the left hand side, we could see a small pocket here. Okay, so that could be a possible hydrophobic site, a possible binding site, right? First of all, I would rotate the molecule, and side by side I would like to tell you what is the characteristics of binding site which could be there. So a binding site, first of all, must follow certain properties. The first property is that it should be accessible, right? the ligand which is binding to the binding site must be accessible. So if I zoom it here, we have certain binding pocket here, but I don't think so. The surface area of the binding pocket is not quite accessible to the ligand, because our benzimidine group is quite a large group, not small either. So it is not accessible to that benzimidine group. I see another binding pocket here. The hydrophobic pocket is also there. So let us explore that particular hydrophobic pocket. Maybe that is a possible binding site for our ligand, right? So the binding site must be having a neither too big nor too small accessible area, the solvent accessible surface area. So the surface area should be large. There should be hydrophobicity. So these are the two requirements currently which we have for our hydrophobic pocket, binding pocket. So in the center, you could see a quite a deep binding site, right? I think that this is a possible binding site where the ligand, which is the benzimidine chemical compound, could be binding with our protein molecule and interacting with it. So let us see, and we have predicted two possible binding sites. For that, we need to delete all of these, the charges, the maps, and all of these measurements. Okay, we need to go to our original molecule. So 3-PTB is our original molecule. Okay, this is the original molecule, and for this purpose, what we need to do is we would just go to our action button, and we would hide everything, and then we would show it as cartoon and color it by the same coloring scheme which we have colored our protein molecule with. Okay, so it will be even now, right? It is even now. You could rotate the molecule. Afterwards, what you have to do is, for the 3-PTB molecule, I repeat that for only the 3-PTB molecule, you have to hide everything, and then you have to show it as cartoon or a surface probably. So this is the surface representation for our 3-PTB trypsin molecule. So let us identify the binding site. So this is the probable binding site which we saw earlier, right? But I think this is quite shallow. It is not too deep. And upper one, we have another possible binding sites. So let us see whether this ligand is binding to binding site 1 or binding site 2. So we have our benzimidine ligand. It is placed in the first amino acid type. So okay, so actually we have quite accurate prediction of our binding site. Let us just change the representation of benzimidine molecule by this show toolbar, right? So we could change the color also to blue, and then we could show it as lines or sticks, probably both will do. So we could show it as sticks. So if I zoom it a little bit, the benzimidine is actually quite fitting in the pocket, right? So after we label it, let me drag it, center it. Okay, yeah, so here it is quite like it is very well fitted into the binding site of that protein molecule, and there must be certain ligands, there must be certain amino acids which are interacting with this ligand. Okay, so now what we have to do is, after the prediction of our possible binding site, we have to identify what are the residues which are interacting with a particular ligand. Okay, so for that purpose, what we could do is, we would use the action button of our protein molecule, right? For the 3-PTB, we need to go to the action button, and then we would perform certain preset ligand binding site options on that particular ligand. Now go to action button, click on preset. You would find an option of ligand sites. Let us see. So this is the option of ligand sites, and the fourth option what we have here is the transparent surface. Once you click on the transparent surface, you could be able to identify the surface and the amino acid residues. Let's click on that. So here we have quite a good representation. It is quite colorful. So this is the possible surface, the binding site, and the amino acid residues which are there, which are interacting. So you could the yellow dashed lines are actually showing the kind of interaction which is happening between among the ligand and the amino acid residues. Okay, so now we are going to select all of the amino acid residues which are there. Make sure you select all of them. Okay, so I just rotated a little bit and all of them are selected. I think something is remaining. Yeah, this amino acid is remaining in the upper portion. Okay, so I would select that also, and one more is there. Okay, so now they are being selected, and see, we have certain interactions, no? And also you could see certain balls there. So these are the spheres. This the we have a small sphere in the background of the ligand molecule, and also a sphere in the outside of the binding pocket. So what are these spheres actually? These are actually the water molecules. So let us now identify these water molecules and what are the importance of these water molecules. See, these are interacting with the ligand, right? So how is this water molecule actually interacting, and what is the importance of water molecules in this interaction? So we have to see. So actually what happens in the X-ray diffraction data, the water molecules are placed so as to properly crystallize our protein molecule. So what we could now do is possibly name the residues. So now we have named the residues here. So the residues which we have here is, I could see cysteine is there, serine is there, right? Alanine is there. Okay, and we also have the glutamine, right? So we have quite a lot of amino acids which are directly interacting. See, we have a aspartate group. The two groups in the aspartate amino acid are interacting with the with our ligand, and similarly is the case for tryptophan, the 215th amino acid. Similarly, serine is also interacting with our ligand. So you could see the interaction, right? And you could just slab your wheel a little and see the interaction of valine, the 227th amino acid residue, which is interacting with the hydrophobic portion, the benzene part of our ligand, right? So these are the interactions and the amino acid which are present at the active site or the binding pocket of our molecule, right? And these are the interactions which are there. So now what are we going to do is, we were talking about the spheres. We have seen what are the amino acid residues interacting, and now you would see in the sequence where are these water located. So scrolling down, we have here a water molecule selected, and what we would do is we would select these two water molecules, right? And we would also now what we would do is we would select these water molecules and then see what is the importance of these water molecules in actually the interaction and the stabilization of these water molecules. Okay, so we would color it possibly by another color, let's say yellow. So okay, the water molecules are being represented here. Let's label them. We could try to label them by the atom name or by the residue name. Let's try each of them. So what do we conclude? The water molecules are not being labeled. This is because in the X-ray diffraction data, as I said, that water molecules are necessary for protein crystallization, but the hydrogen atoms in water molecules are not being diffracted. So hydrogen bonds are not detected, hydrogen atoms are not detected, and therefore we have only oxygen atoms, and therefore this is the reason where due to which the O is written, right? We have a sequence of O, that means oxygen is written in the case of water. So wherever you find oxygen written, that means the water molecule is present there. So okay, so what this water molecule actually does is, we have two water molecules here, and the water molecule which is fitted inside the binding pocket facilitates the interaction between amino acid which is present behind the binding pocket and between the ligand. So it specifies and it actually efficiently stabilizes the interaction between the amino acid residue and the ligand, right? And the water molecule which is present in front of the binding pocket ensures that the ligand is stable and does not come out of the binding pocket. So water molecule is quite important in stabilizing the ligand and protein interaction, right? So that is the importance of water molecules in our current protein analysis study. And so the next thing we have so far we have analyzed the whole protein molecule. See, this is the structure. And now since we have identifying the binding site, we would like to have a high quality image of a binding site which we have predicted. So for that, what we need to do is we need to move our mouse button and play with our sphere. And what we need to do is we need to delete the water molecules so that they do not interact with our molecule in this current session. And what we need to do is we need to just place okay, so this is not being deleted. You could just hide the water molecules. So we hide everything. So after that, you need to rotate and move your molecule so that the binding site is clearly visible, right? So we would just use our mouse, the left click, the right click, the wheel and the move button so that the site is clearly visible. Let's just recenter it. We have to orient it according to our display system, right? So this one, I think this is quite good. The binding site and the amino acid names are also visible. Okay, so now what we could do is after this we would label the residue, the ligand. And now we would open the command line window. Okay, so this is placed here, and now I'm going to open the command line window. Let's see the command line window now. In the command window, what you have to do is go to the menu bar and click on File, and then from the file you could save the image as PNG file. Okay, we have other two options also, but that are not needed for right now. So save as PNG, and then a dialog box will open, and you have to direct yourself to the present working directory. I would name it preferably as first image.png. Yeah, that is a good name, first image.png. So this is the first image which we are producing. Okay, so as we can see, the default resolution of our pixel, the size is 640 into 438 pixels, right? So this is 640 into 438 pixels, but actually the picture is not of high quality. So now I'm going to show you another method by which we could save high quality images. You need to type this command in your command input area, ray. This ray traces the image, and now we need to enter the width and the height of our image which we want to save. So I would specify it in a square box. So I would give the width and height as equal 4000 into 4000. You could specify yourself. So after we have given this, we need to hit the enter button, and then it would be processing our square box and the image will be made, but it will not be saved. So let's hit the enter button and see what happens. So ray 4000 cross 4000 is done. It would take a bit time, and the progress is 37%. The progress is being shown in the command line window. Let us wait for a few seconds. It depends on the speed of your processor. So the image size here is 4000 into 4000 is being displayed in the displayed area. Okay, and now we need to save this image. Click on File and then Save Image as PNG. So the first name was first image, and now it is a second image.png. So you must be wondering that why we have saved two images. Let me show you what is the comparison between the first and the second image. The quality of that image highly matters. So this is the first image, As you can see, the quality is not quite good, and if I zoom in a little bit, it will get blurred out, it will get distorted. So let's zoom in. So you could see, focus on the benzimidine molecule, right? So it is not being zoomed in quite a lot, and it is also getting distorted. Let us now see our second image, which is done by the second method of ray tracing. So this is our second image which we have here, and now focus on the benzimidine molecule. It is not being blurred out to that extent, and we could zoom in more, right? So this is the image which someone would want to publish in your research papers or for your study purposes, right? So the second method is most preferable, I think, for the ray tracing of the image, right? Okay, so what we would do now is we have compared the image, we have seen how to do this, and the second thing we would do is now subsequently open both of these files, the 1U19 and the 3-cap files, in Pymol. Okay, so before that we would need to analyze these files in Notepad++, and therefore I have opened it. What you need to do is, for this particular file, for 1U19, the inactive molecule, you need to delete the chain A. So wherever the chain A is written from atom 1 to the end or the termination of the chain A, you have to select those atoms. You can keep those heteroatoms, no need to delete that, we do not need for analysis purpose. So you need to select all of those atoms, just press on shift and then go to page down button, and then subsequently select all of those atoms in chain A. Because rhodopsin molecule is a dimer molecule, it is a transmembrane protein, so we need only one part of that, one monomer of that, for our analysis or superposition purposes. Otherwise it would become quite messy with the display area. So let us wait till the file is being cleaned up. This is known as the processing or the cleaning of the PDB files. So let us now delete this. We would select it. Find the termination line which is there. It could be written by a TER word, right? So we are now deleting this chain A. Whole of that chain A we have to delete, and we would only keep chain B. As soon as you encounter the ter atom, I think it could be alanine, yes, so this is the ter atom, and now we would delete it. So this is deleted, and now I would suggest you to save it as another file, save as inactive. Since it was this inactive confirmation, you would go to your present working directory, and then from the options, save as all types, and then rename it as inactive.pdb. It would be easier for us to locate it. And similar thing is done for the active confirmation, the 3-cap confirmation. Okay, so the 3-cap confirmation, I have already done that. So this is the active and the inactive form. What we now need to do is, we would now go to our search bar, and for the active and inactive, at one go we need to open that file. So this is we would now close all of the tabs here, because now we are going to analyze it. So we have closed the tabs, and now we will search for our Pymol. Okay, we need to select these two in the Pymol. So we would go to Pymol. Remember, Pymol has many options seen here, but you need to click on only the console option and open it. Okay, find the console and open the Pymol. After this, you have to click on the file option, open, and then you need to direct yourself to your current working directory, which is the desktop Pymol folder, and then by clicking on inactive, pressing the control button, clicking on active, both of the options will be selected, and then you need to open them at one go. So as you can see, these both the files are loaded here. Okay, now what we will do is, we would zoom it. So this is the active and this is the inactive, right? So if I zoom it here, this is the active one. Okay, So green is the active conformation, and if I zoom it in the inactive one with the help of action, the blue one is the inactive one, right? We would display the sequence for both of them, and let's now zoom it here so it is clearly visible. Okay, so these are the two molecules we have here, and then we would need to align these two molecules. So in the command line input area, what we do is we write a command which is the align command, and then the name of the PDB entries. Whatever name is mentioned here, for example, we have active and inactive. So whatever name is mentioned here, be it underscore, be it a numerical digit, whatever there it is there, you need to mention align active and inactive. So it would align or superimpose these two molecules, and then we need to write another command which is all. So by writing all, what does this whole command means? We would align each of the molecule, each of the atom of inactive conformer and active conformer of rhodopsin molecule. So as soon as you hit the enter button, the blue and the red colored patches of protein molecules would be superimposed upon each other. For example, in this, you do not need to worry for the scattered molecules, because these are the heteroatoms. We would not be analyzing them. We would just need the superimposed structures of the inactive and the active conformers of rhodopsin molecule. Okay, so but this wireframe structure is quite messy. So by clicking on the show tab and the hide tab, we could create a more better visual representation, right? So likewise, as we did in earlier section, let's color it by yellow. So this is the yellow one, right? So the yellow is the active conformer of rhodopsin molecule, which is not having any ligand, right? So the ligand is actually retinal, 11-cis retinal. As you can see from the sequence, the yellow colored sequence is the active conformer, right? And the blue colored sequence is the inactive conformer. So we have this entire sequence here. We also have water molecules, but we do not need to focus on that. And so this is our active and inactive conformer. Similar thing could be done for our blue colored conformer, the inactive conformer. Let's do that. We could hide everything and then show it as cartoon. And let's color it by magenta. Okay, so this color is okay. So now what do we see? There are certain regions of alpha helices which are superimposed, but okay, here I see a region which is actually not superimposed. There is quite a lot of conformational change which is happening in the alpha helix region, right? And there are certain portions which are actually superimposed. So what do you gain from this study, from this analysis? There are certain portions of certain portions of molecules which are being affected upon ligand binding and ligand removal. Okay, so what we now need to do is we need to measure what is the conformational change which is happening. So as usual, as we did it in the command line input area, first of all, we need to select the amino acids for which we are going to measure. So I think this random loop will be more relevant. So this pink, the magenta color is for the inactive conformer. Let's select an amino acid from the inactive and the active conformer of the random loop as well. Okay, so this is the active and the inactive conformer. Let's select amino acid. Now I'm going to select the amino acid from the random loops, right? So let's see what the amino acid selected is. So the name of the amino acid, I think probably in this region there would be a serine residue here. So we have selected this residue. What is this residue? First of all, we would just label it. Yes. So this is the 144th amino acid, which is serine. Let's clear everything and show it as sticks, and let's color them by red and green color. Okay, so you have to select only one of them to color that. So this is a red colored amino acid, and this could be a green color. So this thing you have to keep in mind. We have selected both of them. So let's differentiate. Okay, so now this is done. We would label it, and we would try to label the residues. as well as the atom names. So the atoms are clearly visible. We could use our mouse buttons to drag in and zoom in. So let me place it. Okay, now this is clearly visible. So we have both the serine. The upper one, the red colored portion, is of the inactive conformer, the inactive serine, and the red color is the location where the active is being translocated. Let's go to the wizard option and click on measurement, and we would measure the distance between the C alpha atom of the active and the inactive conformer. So let's do that. Let's click on the first atom of the C alpha of the serine of the inactive conformer, that is the red colored conformer, and then we would click on the second C alpha. As you can see, there's a 4.6 angstrom distance or translocation. So what do we conclude by this? Upon ligand binding, upon ligand deactivation, the distance between the active and the inactive conformer actually changes. So we have done this conformation analysis. So that is what the tutorial was all about.